Our first speaker would be Dr. Mona Murray, and her speech would be about tissue engineering, recapitulating the cascade of development.
Thank you very much. And we're honored to be in the BioVision. We hope it will be forever, the BioVision of Alexandria. Today, I will be speaking or throw some lights on tissue engineering, the current status, which we call it recapitulating the cascade of development. The presentation will have three main pillars. One of them is to throw the light on the current approaches, how it takes us from the previous treatment to the current one. The second one is the promise of tissue engineering. And the third one, how is the tissue engineering become a challenging field currently? Now, last century took us from the replacement of a whole leg or a knee toward the synthetic apparatus. And these synthetic materials have grown so much advancement during the past century. However, it's still artificial. And it's not functioning the same. If you see that this is a knee replacement, and the revision of it is very difficult. Now, currently, years ago, we used to make replacement therapy for old people. But now, due to the huge accident occur, wars in the young people, we are faced with young population with a lot of disability. Now, is that correct to give them the artificial synthetic apparatus? No. It does not last. It does not give them the accurate function they need. They grow. It does not grow with them. That's why the disaster of losing them, as you see, when we have a hip replacement and a fracture happen, people lose bone and lose the head of it. And it becomes very invasive procedures after that. Now, how do you see the improvement? It's still synthetic, and it does not work, and it does not help the growing child and the growing adult if he lived for 60 years after that. Thank you. It is still synthetic. It's not natural. And what will happen if they lose this? They go into severe disability and society problem and a huge amount of money impact in this. And even the replacement therapy become huge, so the disability grows with it. In the last century, the last 10 years of the past decade, they introduced the field of regenerative medicine and tissue engineering. Now, in this field, we have the promise of stem cell therapy, where you can isolate stem cells from the body, seed it on a scaffold, return it back to the patient to have what we call it living tissue for the patient. Now, the promise of it was huge. When we started this field, we thought it's the end of the therapy. We will face everything. The challenge was so huge. You can take a biopsy from a patient, grow like a spot of blood or a bone marrow, and grow cells in the lab. Of course, I'm simplifying the things here. And we were succeeded several times to grow pieces of bone in the lab. Did this solve a problem? Yes, to some extent. Diabetic patients were able to have relief of their ulcer from there. Here, you can see the treatment. Yes, it makes a lot of improvement. It does make skin tissue engineering, using cells from the patient or a biopsy from the patient, transplanted back to the same person where they can grow the cells. Look at the advancement through the past decades, a lot. We're starting from this artificial and this scarring of the patient until we reach here. Yes, it was a lot of reward come from this side, the cell therapy. which we can isolate cells from different parts of the body. This is a huge field. I'm only throwing some lights on it. It grows more to make organogenesis. So they were able to make trachea from the cells of the patient that transplanted, and they made a whole organ out of the cells and a scaffold, exactly carbon copy the shape of the original organ of the patient. Now, this is a successful thing? Of course. It helped a lot of patients. For example, the limbus stem cells from the eye, they can take a biopsy. For the people who doesn't know this, the depths of this thing, isolate the cells, put it on a collagen scaffold, return it back to the patient. Yeah, a number of cases have gained successful sight after that. We agree 100%. The successful of it was so much, we were so fascinating of the field at the end of the last decades and the beginning of this one. Now, also arthritis? Yes, of course. They remove the cartilage, they replace it with cells and the scaffold, and it made a lot of progress for patients. So this is the reward of it. However, like everything else, it goes from one generation to another. The field is so expensive. If you happen to need 40 million of cells in the lab, you can grow them for 3 weeks. You will sit next to them. You will need media and people dedicated. It's expensive and dedicated and high technology for it. And what will happen if these cells die or get infected? What will happen that you will die next to the patient? This is a huge dedicated field. Yes, it succeeded a lot, but it has other things. Back in 2004, Assay have published a beautiful article saying, my goodness, these companies have bankrupted already. Why? What happened to the product of tissue engineering? We sat down, all of us, including the NIH and NSF and all the society in the world, to find out why that tissue engineering and stem cell therapy does not work with everybody. Aging, very important. If we rely, this is number one, if we rely on cells, it's impossible. The cells from the embryo is definitely different from the infant and is definitely from the adult and the aging population. Not only, if you take a biopsy with the same size from different people like this, now, and you isolate the cells, the number of cells is different. As we grow older, it becomes less. The ability of these cells to divide and proliferate becomes less. So we cannot make a therapy for young people only. Not only this, stem cells, when they grow, they make colonies. These colonies different in numbers and in size from one person to another. It depends on the age, the health. So we cannot just face our life on this. This is number one phenomena. Number two, we were faced that these stem cells that we kill ourselves to move it from bone marrow and peripheral blood, it's everywhere in our body. Wherever you have circulations and you have small capillaries and arterioles, these nice parasites are hanging around, and these are the stem cells. We didn't know that 20 years ago. We only knew this 7, 8 years ago. That's why we start thinking different. If God gave us all the arterioles and the capillaries in our body, and we have these parasites that they are stem cells hanging on them, why not? We cured from inside. Why do we have to transplant cells? Now, what is exactly if we transplant the cell therapy does to the body? It turns to be that these parasites or stem cells move during injury. And they have the paracrine and autocrine. They send signaling everywhere. They move like a drugstores to relieve the body. But are they enough? Not enough if they are in an older age. And although this happened, Look at how Europe is the only one has multi-centers. Of course, it's not there in the United States because of the FDA regulations. Look how much is the success of the cell therapy and tissue engineering published until, this is at 2011, and published till the survey of 2009. Yes, a great thing. And how many countries? It's all Europe, exactly, are working in cell therapy. But are all the cases successful? Yes. When the patient is young, it is successful. When the patient is older, it has a question mark. Another third phenomena here we discovered, that stem cells, when you inject it in the body, it lives only for 3 weeks. What? Watch this. 3 weeks. Millions of researchers have done on this. It dies. It sends signaling everywhere, and we could capture it necrotic in different parts of the tissue while the tissue made the bone. Why? Because it sends signaling for the own host cells to work. We started from this point to think differently. If cells make the tissue, we call it environment. We call it niche. This niche in a scientific matter is made by the cells, and it's the hallmark of cells. If cells wants to make muscles, it will make the niche of the muscles that has the characteristic of the muscle with its tensile and elasticity. If it will make the bone niche for the bone, it will have the strength of the bone. So this niche is made by cells. Why not we think differently? And instead of working on the cells, let's work on the niche. When we grow this is the skin with the cells. When we grow that in the lab, this is originally a stem cell that you leave it to grow until it makes the niche of it. So maybe the niche is the thing. We start discovering, and all of us around the world start learning and studying more about the niche that cells grow. And we found when we see cells on a 3D, it's totally different from the cells on a sheet thing. Not only this, look at the fibers, the nanofibers. Cells, when they grow on the nanofibers, they divide in front of you. You can see it in the microscope live. And when you leave it for a few days, it goes underneath and it wrinkles, making a tissue like that. So if it's the same cells, it makes different. We started thinking differently. Let's study how is the tissue organized here. This is a piece of the intestine here. We took it and we decellularized. We killed the cells in it in a special protocol that is well known. Observe the niche the cells make. Some of it is nanofibers. Some of it is large fibers. Some of it is macro and micro molecules. We start freeze drying this to make it as a material, and we found it very strong. Watch and observe. It's not stationary. We used to make the scaffold for the cells to make the bones and cartilage in a special stationary form. This is not what God created. God make it irregular, individualized. You have all kinds of tissue architecture and tissue strengths and topography in it. It turns to be another thing that elasticity of the tissue differentiate the cells. So cells secrete the niche and the microenvironment, live in it. Then this microenvironment, if it's softer, it will make the cells neurons. If it's a bit harder, it will make it muscle. If it's more harder, it will make it bone. Amazing. We never think of it. We start to change to the second part of my presentation here, to biomimetic tissue, to look at life sciences surrounding us, how God made this tissue and to imitate it as much as possible. Not only in topography, on architectures, on a 3D, in chemistry, in behavior per time. We start thinking, why not we work on our scaffold to make cell homing, to use the endogenous stem cells, Let's not work in the lab anymore, use the lab only to study the phenomena, but go back to the host. Make something inside the body that will attract its own cells. Now, of course, that's much easier. Is it happening in nature? Yes. Homing during injury, it happens. Cells already have signaling molecules in it that it rolls and reaches the injury site. So it is occurring through what? Your blood circulation. Now, of course, this is a huge field. I'm only scratching some of it here. Now, based on this phenomena, everybody, everyone in the whole world start working to home the cells. They start decellularizing the heart from the cadaver, use it as a scaffold with this beautiful architecture, and then study in the lab if they seeded with cells, what will happen. It did work. In our lab, we bring the liver, and we decellularize the loop of liver to study the architecture of it. And it's a beautiful documentation in our lab and in many other labs that when you remove the cells, you found architecture that none of the synthetic scaffold or biomaterials can do it. That's why when you seed it back with cells, they make liver, all right? So it's homing. Cells go based on architectures. It has its endogenous signaling that it makes it aware of the media. Now, another example. Years and years, we used to isolate cells from the adipose tissue. Currently, we think differently. We kill the cells from the adipose tissue and make a scaffold that the cells grow on it. So the field this can be work for cartilage with its. Now, homing, shaping, it's not only the architecture, the shape of the synthetic material, the architecture of it inside can home, you can put a whole scaffold, look like a tooth, and then you implant it and bring a whole tooth structure after that. Now, it's recapitulating nature. It's what God made. Look at the nature surrounding us. We start taking things from K to Z and making. Now it's already in the market. People take the blood from the patients and centrifuge it and make these skin mask where they can attract cells, and they start making face looks younger. Now, and it has a lot of application. One of the most things that's growing heavily in our lab now is to decellularize the cells from the bones and keeping the architecture of the bone. Now, look at the bone. What does the bone has? It has several trabecules, and it has collagen and hydroxyapatite. And it's moving. Now we prepare that, and it will be produced soon by Farkov, a European pharmaceutical. Now, a case here just to show you how beautiful. Cyst on both sides, we remove the cyst and we put the hydroxyapatite, and you can see the difference. Now, we home the cells, even if it's a large area. Now, another example, quickly here, we prepare bioglass, but we prepare it with the architecture of bone. Remember, the bone has a transverse and vertical trabeculae. For the first time, it's prepared like the bone. Has trabeculae that when it's degraded, it becomes thinner and thinner. Cells recognize it. We are able, look at this beautiful, cells recognize the architectures like a bone and start homing to make bone here. Now, another thing is the topography. I'm quickly going through this. This is a current dental implant that they make a lot of failure because it's not recognized by tissue, by cells. We knew that cells like topography here. We start aligning aligning by making a grooves with nano ablation here. Cells recognize it. And when we grow them on a smooth surface, they look like this. On a laser surface, they look like this. And we RGD, they coated and aligned between the groups. So if we put this in patient here, and we take it from the tissue, and we removed this. We did not put cells. We found the cells of the own body aligned. These are the cells, they align. They recognize surfaces. They produce octacalcium here, which is the tissue that will adhere to the dental implant. Homing is the current, this is already in market right now, and this is the patient. Again, we're recapitulating development. Thank you.
Thank you, Dr. Mona, for sticking to the time. We will leave the question until the end of the session. Our next speaker will be Professor Hassan Azazi, and he is going to talk about gold nanoparticles, a versatile platform for sensitive biosensing. Please welcome Dr. Hassan Azazi.
Assalamu alaikum. Number one, I would like to thank the organizing committee, Dr. Ismail Saraguddin and Dr. Mohammed Al-Faham, and the staff of Bibliotheca Alexandrina who coordinated my talk to you today. I'm very pleased to speak to you today, and I hope that you will find my talk useful, especially for the young scientists and faculty members who are either doing research or want to start research. The topic of my talk today will be on gold nanoparticle, a versatile platform for sensitive biosensing. This is the American University in Cairo, and for those of you in the sciences and engineering, we have a large number of graduate programs in biotechnology, nanotechnology, food chemistry, and several other engineering specialization. And I encourage you to visit the website of the American University in Cairo, and if you're interested, you can apply to get your master there or PhD. Before I start, I would like to recognize the real players, the people who have actually done the work in the lab, and they have a long army of students, and this is not all of them, but I would like to recognize students who have really done and contributed to this work: Sharif Shawqi, Tamir Samir, May Mansour, Salma Tamam, Ahmed Nasir, Rima Olabi, Basim Gergis, Kamil Eid, Marwa Hussein, Hiba Adil, Fatin Farouk, and Karim Abdel Hadi, and Shiri Daniel. We have a large network of collaborators in Japan, Dr. Sharif Safti, in Portugal, Lisbon University, Dr. Ricardo Franco, Fra University, Dr. Derek Bald, Dr. Sana Isa from National Cancer Institute, Steve Kazmikizak from Oregon Health State University, Balhorn from UC Davis, and Dr. Shahad from Suez Canal University, and Dr. Altani from Qatar University, and of course, Masal Khair. These are pictures of my students in the lab. And I would like to recognize this group in particular for their effort in actually winning the first place in the Middle East and third place on the globe in technology business plan competition. My talk today will be different. We are trying to be innovative in the way we do research. We utilize high technologies like biotechnology, nanotechnology, and medical technology to develop yet low-tech medical innovative solutions. So we're trying to overcome the need for very large budgets and complicated research that require very sophisticated lab to yet develop, again, solutions that are innovative but low-tech and can really contribute to the society. We have developed a nanogold assay prototype for direct detection of Hcv Rna, and we'll talk about this. But we also have expanded the platform, and we have developed additional 8 different platforms. I have spoken in BioVision in 2010, and gave a short lecture on gold nanoparticles and their diagnostics and therapeutic application. And what I'm going to present today is the work for over the past 2 years. Before we start, I believe nanoparticles and nanostructure is a very rich area of research. I believe, as Egypt, may have missed the biotechnology revolution, and we did not contribute much to the biotechnology revolution. However, I believe there is still a chance for us to catch up with the nanotechnology revolution. There is lots of areas that we can contribute to, very fertile environment for doing research that is not necessarily very costly if you think innovative and try to use things that are suitable for your budget and your lab infrastructure. Nanotechnology offers us a large number of tools in terms of different structure. If you go to the nanoscale, which is 1/1,000,000,000 of a meter, you can see that you can form a structure that can differ in the nature of the element itself. You can have gold or silver or iron or platinum and so on. Usually the size between 1 to 100 nanometer. The shape, you can change the shape to spherical, rod, flower-like, stars, and so on. You can change the charge on the surface of these nanostructures. You can do chemical modification to add certain chemical group on the surface, and you can do biofunctionalization by addition of DNA molecules or antibodies or proteins. All of this we can do in the lab. So for a particular biomedical application, we can select a nanoparticle with a particular size, particular element, particular shape, particular charge, and particular surface modification or biofunctionalization in order to meet or achieve this particular goal. So in this case, there is a very large number of opportunities that you can develop a different structure that can perform a very important biomedical value or function. These are different shapes of gold nanoparticles. You can see nanospheres, nanoshells, cubes, nanorods, nanocages, and nano triangles. You can see on the side here, this is actually a vial of gold nanoparticles that we prepare in the lab, and this is the color of gold nanoparticle in solution. You can see gold nanoparticle in solution when they are monodispersed and suspended in solution, they have bright red color. Why did we select a gold nanoparticle? Because gold nanoparticles have very unique property called surface plasmon resonance, where the electrons, the conduction electrons on the surface of the metal oscillate in response to electric field. In this case, will be light. And this property happens only if gold nanoparticles are greater than 2 nanometer and around maybe 100 something nanometer, and the bulk gold nanoparticle, or the bulk gold does not exhibit this important property. You can see these are different vials of gold nanoparticles. When you change the size, you can see that the color of the solution change when you change the size of gold nanoparticle. Gold nanoparticles also have an interesting property that we observed in the lab, and actually reported by many others, that when gold nanoparticles start to aggregate around the target, their color change from bright red to bright blue. You can see. And we have utilized this very simple property to develop a new test, a novel test to detect hepatitis C virus RNA in serum of patient using a colorimetric assay based on observation of color change and without the need for sophisticated instrumentation. As you know, to detect Hcv RNA in serum, you require RT-PCR, and this is very expensive, 300 to 600 pounds. In addition, that you need sophisticated equipment, you need highly trained technician, you need complicated infrastructure for the lab. All of this we have developed a strategy to do it in a simple way, yet effective way, and also much cheaper, 1/10 of the cost of current assay. Gold nanoparticle can be used in different strategy. Again, remember, for any nanostructure, you can change the element, the size, the shape, the charge, the biofunctionalization strategy, and at the end, you can develop or construct a new structure that can achieve a particular function. Gold nanoparticle, in addition to their ability to change color from red to blue upon aggregation, they also have the ability to quench fluorescence. And we have developed another assay to detect also Hcv Rna, but using a different strategy. In this case, we form this probe of gold nanoparticle and a fluorophore. The fluorescence from this fluorophore will be quenched by the presence of gold nanoparticle. But in the presence of target DNA, in this case will be Hcv Rna, this probe will open and hybridize, and the gold nanoparticle will be separated from the fluorophore, which will fluoresce. So if we see fluorescence, we know Hcv is in this serum sample. This is a different property of gold nanoparticle, and the difference between this gold nanoparticle and the previous gold nanoparticle is the size. The nanoparticle that change color from red to blue are around 15 nanometer. This is between 40 to 80 nanometer. So changing the size will give you a different property, a different application, different strategy to develop your technology, whatever you want to do. Gold nanoparticles can be synthesized in the lab very simply and very cheaply. This is nothing really, there's no rocket science here at all. There are many methods established to synthesize gold nanoparticle in the lab. You can choose this method to synthesize something between 0 to 5 nanometer, this method to synthesize between 60 to 50, or 50 to 147, or 50 to 200, depends on the method that you want, on the size. You can select the method for synthesis. If you want to know the equipment needed to make gold nanoparticles, you will be surprised. This is what's needed. Who does not have this in the lab? We need a buffer. Oh, this is gold tetrachloride, by the way. 1 gram of gold tetrachloride, which is enough for over 10,000 tests to detect HCV, is 200 euros, which is essentially 1,600 pounds. طبعا يعني very low price. Need a flask, and then we reduce this using trisodium citrate, and then the color of the solution change to blue, to red. Here we have gold nanoparticle. We do some characterization. If you do not have this sophisticated instrument in your school or your faculty, you can really do this in any of the collaborating institutions in Cairo or Alexandria. And this is some characterization using scanning electron microscope. You can see monodispersed gold nanoparticle and their average size. As you know, hepatitis C virus, when it infects the body, we have acute infection. Within 6 months, if the virus persists, this will change into chronic infection, and the antibodies start to rise and then stay in the body. And then the Hcv Rna either can go down in case of acute infection, and if the body able to resolve the infection, then the Hcv Rna will be cleared. If the virus persists, I'm sorry, this is not working. If the virus exists in the body, then we have a chronic infection, a patient with infection. If the liver enzymes change, this patient may need to be treated. As I mentioned, current Hcv testing in the market happen in 2 stages. Number 1, an antibody test, and then number 2, Hcv Rna test. The reason that we do antibody test first is because it's cheaper, between 20 to 40 pounds. But this is, if the result is positive, this is not conclusive that the patient is actively infected with the virus. And we need to do a molecular testing in order to detect the presence of Hcv Rna. Then, if the result is negative, this does not mean that the patient does not have HCV, because he may be immunocompromised, or maybe he has been recently infected and the immune system did not have enough time to develop antibodies. As I mentioned, we have offered our Hcv nanogold solution that can detect Hcv Rna. For our estimation is around 50 pounds, which is almost 1/10 of the cost of the current technology. Our run to the assay itself, you can see we can get a serum sample, and then we extract RNA using any of the existing technology. And by the way, we have developed our own silica nanoparticles coated with oligonucleotide for Hcv Rna that we can specifically isolate RNA from the serum for HCV. Then we add gold nanoparticles, which are red, and a short DNA sequence called oligotargeter. This oligotargeter really direct, or is specific for a conserved region on the genome of all HCV genotypes, which is very specific and can detect any HCV genotype. If the virus is there, the oligotargeter will hybridize to the RNA target, and then the color of the solution will change from red to blue, because we have constructed the chemical environment in the hybridization mixture that in presence of the virus, the chemical environment will force the gold nanoparticle to aggregate and give us a colorimetric signal. As you can see, the difference between an HCV negative serum and positive serum. We have done many clinical trials, and we have very, very good sensitivity and specificity of this developed prototype as compared to real-time PCR commercially available in the market. As you can see the difference, this is just to give you a quick comparison. We have a major reduction in the cost, 1 step versus 2 step, less than 1 hour to generate result, simple method, basic infrastructure, and entry-level technician. We have filed 4 patents to protect the technology with the USPTO as well as PCT. We have been invited to give many presentations and have been published several things. We have been cited in Science magazine last year, and also in Nature magazine, and also Nature Middle East last year for developing such technology. I try to push the envelope a little bit and encourage my students, so I formed a team, and we entered the Technology Business Plan competition that is organized by the Arab Science and Technology Foundation. 240 teams enter the competition in all fields, computer science, network, energy, environment, food, you name it. And we have won first place that was held in Bahrain. The competition, the final stage of the competition was held in Bahrain. We qualified also to compete with the Intel Global Challenge that is organized by Intel and UC Berkeley in the Silicon Valley in California last November. And for the first time, an Egyptian team or any Arab team wins a third place in this 7-year long competition. My challenge to you today as a young staff and young researcher is to enter the Arab Science and Technology Foundation competition this year. You need to form a team, you need to have an idea, and you need to submit a proposal and a business plan and plan to develop a prototype of whatever research you are doing and try to enter this competition. This is a great learning experience. You will learn a lot. You learn how to develop a business plan. You learn how to focus your research on a societal problem or societal pain that you can solve or contribute to. This is from the Arab Science, and this is from Intel Global Challenge. And by the way, we have formed a company, and we are about to spin off American University in Cairo. American University in Cairo has been in existence for 93 years, and this may be the first spin-off company of AUC. We have tried to expand the technological platform, and we have now moved toward detection of cancer biomarker, and we have one student working on detection of urinary bladder cancer and hepatocellular carcinoma, another one, and also cardiovascular disease marker. We have developed a quantitative FRET or fluorescence resonance energy transfer nanogold HCV assay that uses this technology. The technology here is different. Again, every time you change the size and you change your strategy, there is essentially countless ways of how you can do this. Not only for detection of disease, but for doing whatever you want with regard to nanomedicine. How many minutes? 5 minutes? Okay, thank you. So I have to fly. We have also working also to change the shape of gold nanoparticle from spherical to nanorods in silver nanoparticle. And this is the shapes of silver nanoparticle that we can make. We can make silver nanoparticle any shape or size. And these have very important applications in wound healing and antibacterial effect. As we know, silver nanoparticle have very strong antibacterial effect. We have developed another assay prototype for detection of tuberculosis. Tuberculosis kills 1.3 million people, and we have developed an assay using gold nanoparticle to detect DNA of tuberculosis in clinical specimen. I'll not go into details. This is just to give you the result. We have also developed another assay platform for detection of C-reactive protein, which is a marker of inflammation, and a marker can be used in the assessment of or management of cardiovascular disease patient. This is a different strategy. Here we have a competitive fluorescence immunoassay when we label the antigen with fluorescence, and we have the gold nanoparticle with antibody immobilized on the surface, where the antibody will compete for CRP in the serum of the patient and CRP that is labeled with fluorescence, and we can form a curve and you can see we have a very linear response from 0 to 10 nanogram per milliliter, which is very sensitive, by the way, comparable to existing assay. This is also a CRP. Finally, we have developed another assay for detection of malaria, and this has been done in collaboration with the Lisbon University in Portugal. And you can see we have formed here a different strategy to detect a malaria antigen. Again, the monoclonal antibody is conjugated to gold nanoparticle, and we have the labeled recombinant Plasmodium falciparum HSP or HCHAC protein 70. Again, a competitive immunoassay for detection of malaria, which is also, by the way, very important and encourage researcher. This is a very hot area of research, and there is a great deal of funding for malaria research, and encourage you to consider this if you can. In conclusion, we have developed several platforms for detection of many pathogens and infectious disease, but we also have other areas of research that I will scan through quickly. We are interested in identification of targets, and we have done so for hepatitis C virus. And you can see here a dimer of E2 protein that the virus used to enter the cell, and we have identified conserved sequences at the dimerization site that if we prevent, we will be able to inhibit the virus from entering the cell. We are also interested in drug design, and we are interested in formation of what we call shells, or selective highly affinity ligands. We are interested in drug delivery using smart nanocarriers, and we can control the release of the drug and also targeting these nanoparticles to enter a cellular location like the nucleus. This is another form. Here we're using transcription factor to target the nucleus, to reprogram the cell to make them stem cells, and we can use them for multiple applications. Thank you very much.
Thank you, Dr. Hassan, for sticking to the 20 minutes again. And congratulations for the 2 prizes.
Thank you.
We hope that in
Yes, all of them.
the next BioVisionAlexandria will come up with
many.
some good news.
We have many of them.
Our next speaker is Professor Patrick Couvreur, and he will talk about nanomedicine for cancer treatment and diagnostics. Please join me in welcoming Professor Patrick.
First of all, ladies and gentlemen, thank you very much for the invitation at this BioVision conference, to giving me the opportunity to deliver a short talk on the use of nanomedicine for cancer treatment and also diagnostic. As you know, probably the conventional cancer chemotherapy is hampered by drug resistance due to physiological barriers, but also to drug resistance at the cellular level. Also, the conventional cancer therapy is limited by a non-specific biodistribution, by a rapid biotransformation, metabolization, leading to a rapid clearance from the body. And in development, the construction of nanomedicine can be a solution to all those, not all those problems, but some of those problems. Indeed, if you are encapsulating an anti-cancer drug in a nano device of around, let we say, 100 nanometers, and if the encapsulation is well done, you will be able to protect the drug from the degradation. If you are able to construct a nanotechnology which is in fact decorated by specific ligands which are able to recognize some receptor or some markers at the level of the cancer cells, you will get an efficient targeting towards cancer tissues and cancer cells. Also, you know that you have a lot of anti-cancer compounds which are unable to diffuse spontaneously intracellularly, like the nucleic acids. And in that case, after association with a nanotechnology, you will induce a better intracellular penetration of the drug because of the endocytotic process allowing the nanoparticle to deliver the drug intracellularly. Also, as I will show you, the use of nanomedicine will allow, in certain cases, to overcome the so-called cancer resistance to the treatment. And the last but not the least, if you are able to construct so-called multifunctional nanomedicine, you will be allowed to combine an imaging functionality and a therapeutic functionality, which is called now the nanodiagnostic. And I will try to illustrate all those points through very rapid example of technologies arising from my research group. The first technology is the development of the biodegradable polyalkyl cyanoacrylate nanoparticles. In fact, those nanoparticles, after intravenous injection, will concentrate into the liver. Why? Because, in fact, after intravenous administration, those nanoparticles will be opsonized. This means that you have a lot of blood proteins, including opsonins, which will adsorb at the surface of those nanoparticles. And this is the reason why the Kupffer cells of the liver will be able to catch those nanoparticles. So it is really a very nice navette to deliver a drug into the liver tissue. And we have taken advantage of this liver distribution to use those biodegradable polyalkyl cyanoacrylate nanoparticles to treat the multi-drug resistance hepatocarcinoma. In fact, you know that a lot of cancer cells, including the human hepatocarcinoma, are expressing, as you can see here, efflux proteins. And those efflux proteins will be able to efflux the drug from inside to outside of the cells. It is really a natural detoxification mechanism that the cancer cells is using to fight against the anti-cancer treatments, for instance, with doxorubicin, which is a typical anti-cancer compound. And you can see that if you are incubating increasing concentration of doxorubicin as a free drug, you can see that you will be unable to kill the human hepatocellular carcinoma. So the idea we had a couple of years ago was to encapsulate the doxorubicin into those biodegradable nanoparticles, in fact, to mask, as you can see here, the doxorubicin from the recognition by the P-glycoprotein. And it is working, because as you can see here, after incubation of the same increasing concentration of doxorubicin, but as a nanoparticulate formulation, you will completely restore the sensitivity of those hepatic cancer cells towards the doxorubicin treatment. And as you can see here, this was fully confirmed by using a hepatocarcinoma in transgenic mice. You can see that only the doxorubicin loaded onto the polyalkyl cyanoacrylate biodegradable nanoparticles were able in vivo to induce the apoptosis of the cancer cells, which was not the case after the treatment with the doxorubicin-free. And this is the reason why we have started with clinical trials, and to do that, we have created a startup company which is called BioAlliance, and we performed the first phase clinical trial and then a second phase clinical trial, and now the second phase clinical trial is finished, and we started a third phase clinical trial. And you can see here the summary of the results of the second phase clinical trial. After, as you can see here, treatment with the doxorubicin loaded onto those biodegradable nanospheres, we get a survival rate of almost 90% of the hepatocarcinoma-resistant patients. And with the conventional treatment, we get only 50%, 54% survival. And you have here a very nice image, but my pointer is dead, I think. And you can see here very clearly that before the treatment, you have the cellular hepatocarcinoma that you can recognize with in red. And 4 weeks post-treatment, in fact, you can see very clearly that you get a strong necrosis at the level of the hepatocarcinoma, showing that you have a decrease in the tumor size and an efficacy of the treatment. Now, what we are doing more recently is to develop, based on those biodegradable polyalkyl cyanoacrylate nanoparticle technology, a versatile and efficient platform by constructing multifunctional nanoparticles. And those multifunctional nanoparticles are based, as you can see in blue, on a biodegradable polymer core. By using the well-known click chemistry, you can link, you can click, as you can see here in pink, in fact, rhodamine, a fluorescent dye, which will be linked to the core of the nanoparticles. You can decorate those nanoparticles with polyethylene glycol in order to avoid the opsonization process, to avoid the liver recognition, and to go in other area of the body. And finally, as you can see, but again, my pointer is not working, you can see that you can decorate those nanoparticles with some specific ligands like biotin. And you know that at the level of the cancer cells, you have some biotin receptors. And if your nanoparticles are decorated with biotin, clearly you are able to recognize in a highly specific manner the cancer cells. And you can see that on those confocal microscopic studies. In fact, you can see very clearly that our biotin-decorated multifunctional nanoparticles were loaded with a red fluorescent dye rhodamine. And if you are incubating those nanoparticles with the MCF7 breast cancer cells, you can see the second line that the cancer cells will become red. This means that the particles are able to target the cancer cells and to release the fluorescent dye intracellularly in a highly specific manner. Now, the line above, you are using the same nanoparticles, pegylated, but without the biotin ligand, and in that case, you can see that you have only the green color of the actin fibers of the cancer cell, but not the blue color. This means that those nanoparticles were unable to recognize the cancer cell. So by using those multifunctional nanoparticles with specific ligands, you can see that you can have a highly specific treatment of cancer expressing some receptors. So this can convince you that there are really impressive progress which have been made to design, to construct disease-targeted nanomedicine. But actually, on the market, you have only, I would say, depending on what you are counting, 5 nanomedicine on the market, which is poor if you are looking to the high number of publications which are published throughout the best international journals. And this means that there are still very important limitations. The first limitation that you can see there is the burst release. The burst release means that, I would say, a fraction of the drug which normally should be encapsulated into the nanomedicine is just located at the surface of the nanotechnology. And this fraction of the drug, after administration of the nanomedicine, will be immediately released into the bloodstream before to reach the cancer cells or the cancer tissue. This is what we call the burst release. And therefore, you have an inefficient treatment of the cancer cells if you have a high burst release. The second limitation is what we call the drug loading. The drug loading is easy to understand. It is the milligram of the drug, of the biologically active molecule, which is transported by milligram of drug transporter material expressed in percentage. And in fact, if you are looking to the classical nanomedicine technology, including those polyalkyl cyanoacrylate nanoparticles, you have just a few percent drug loading. This means 1 or 2% drug loading. This means that when you are injecting 1 milligram of the anti-cancer drug, you have to inject 100 milligram of the transporter material. And this is a big problem, because either you will not reach sufficient concentration at the level of the target cancer cells, and therefore you will be inefficient, or you will have to inject high number, high quantity of transporter material, and then, of course, you can have toxicological problems. And therefore, there is actually a clear need to develop bio-nanomaterials to overcome those two limitations. And this is what we did now very recently by using a biomimetic approach that we called the squalenoylation. In fact, as you can see, the squalene is a natural biocompatible lipid that we have everybody in our body, especially at the level of the skin. And you know that in the mammalians, the squalene is a precursor of the cholesterol's biosynthesis. And to be transformed into lanosterol and then into cholesterol, the squalene has to adopt a dynamically folded molecular conformation in aqueous solution. And in fact, we have taken advantage of this compact, dynamically folded conformation of squalene to link the squalene to small anti-cancer or antiviral molecules, especially nucleosides analogs, to get nanoparticles. And in fact, we have chosen the nucleosides analogs, first of all, because in general, those molecules are rapidly metabolized. For instance, the gemcitabine that you can see here, it's difficult without the pointer, But the gemcitabine here, when you are injecting this anti-cancer compound is immediately deaminated into the bloodstream by the blood deaminases. So as you can see here, we have linked the squalane with the amino group of the gemcitabine to protect the amino group from the metabolization, from the biodegradation, from the metabolization in an inactive metabolite. And we get, as you can see here below, the so-called gemcitabine squalane bioconjugate. And when you are putting that molecule into water, you will get a spontaneous self-organization of this biomolecule as nanoparticles that you can see over there. And if you are looking to this picture, you can see that you have a supramolecular organization that you can investigate by X-ray diffraction at the synchrotron. And in fact, we observe that we get an inverted hexagonal structure. And the particles are around 100 nanometers. And it is really a platform, because you can see that we have applied this technology to other nucleosides analogs, including the anti-Hcv compounds, which are the DDC, the DDI, or even the AZT. And because those molecules have a poor intracellular diffusion, they are inducing resistances, the linkage to the squalane, and the obtention always of nanotechnologies will allow to improve the treatment. And also, as you can see here, 5 minutes, okay, it will be short, because I have not the pointer, so it takes a little bit more time. So you can see that if you are calculating, for instance, in the gemcitabine squalane bioconjugate, the molecular weight of the gemcitabine, which is the biologically active anti-cancer compound, and the molecular weight of the squalane, you can calculate that you have a 50% loading instead of the 1 or 2% loading. And you have no burst release because you have a chemical linkage between the transporter and the squalane with only an hydrolysis and a release of the active compound intracellularly. Now, what happened in vivo? In fact, in vivo, we tested the efficacy in 2 different models, the L1210 murine leukemia and an orthotopic human pancreatic cancer. You can see on the left side that if you are injecting, in fact, the cancer cells intravenously, you will have very rapidly spread of the cancer cells in the major organs with a lot of metastasis. And in the blue line, you will see that the untreated animal will die after 20 days. So you can see that if you are treating those animals with the GemSAR, the gemcitabine as a free drug, which is on the market actually, at the dose of 100 milligram per kilo body weight, you will increase a little bit the lifespan of the mice because they will die only after 40 days, but you have no long-term survivals. On the contrary, if you are treating the animals with the nanoparticles, with the gemcitabine squalane nanoparticles at a dose of 20 milligram per kilo body weight, 5 times lower dose, you can see with a red line that you have 75% of the survival of the animals and a decrease, an absence of any metastasis. And the same was observed in the human pancreatic cancer, which is an orthotopic model, the cancer cell being grafted into the pancreas. And also you can see that you get a very strong decrease of the size of the tumor after treatment with the nanoparticles, and also that you had long-term survival. So remember, we have used here a dose of 100 milligram per kilo body weight of gemcitabine. Now, we have tried to improve still the efficacy of those nanoparticles by constructing magnetically guided nanoparticles. And you can see that above on the left side, we have used ultra-small iron oxide nanoparticles in in black, as you can see, and in white, we have nanodeposited the gemcitabine squalene at the surface of those nanoparticles. And because the ultra-small iron oxide nanoparticles are magnetically sensitive, if you are focusing a magnetic field at the level of the tumor, you will attract those nanoparticles with their gemcitabine content in a highly specific manner into the tumor. And you can see that it is working. Here, we used a dose of 5 milligram per kilo body weight instead of 100 milligram per kilo body weight. It's not homeopathy, but I would say almost homeopathy. You can see that all treatments failed, but on the contrary, with a black square, if you are injecting the magnetic nanoparticles of gemcitabine squalene and inducing a magnetic field at the level of the tumor, you completely suppress the tumor growth for a dose as small as 5 milligram per kilo body weight. Still more interesting is the fact that, as you know, the iron oxide nanoparticles are able to induce a decrease of the T2 signal at the magnetic resonance imaging. So you can follow the efficacy of the treatment, as you can see here on the right side, by looking to the image after the treatment. You can see, sorry again, on the top, that if you are not focusing a magnetic field at the level of the tumor, after 20 days, you have a tumor with a relevant size. And in B, now if you are focusing the magnetic field and injecting the magnetically guided nanoparticles, then you can see very clearly the collapse of the tumor. And this is a concept of the diagnostic, because in the same nanotechnology, in the same nanodevice, you can combine both a therapeutic functionality, because you are curing the tumor, you are treating the tumor, so a therapeutic functionality, and an imaging functionality due to the iron oxide nanoparticles. And before to finish, I would like just to mention another example that we have recently published in Nature Materials concerning nanodiagnostic using hybrid nanoparticles of metal iron oxide frameworks, what we call nanomorph. In fact, those nanoparticles are hyperporous, and by using different organic ligands, you are able to construct pores with a size corresponding exactly to the size of the invited drug. And in addition, because those are iron carboxylates frameworks, the iron is able, as you can see over there, to induce also a signal at the magnetic resonance imaging. So again, by using this type of multifunctional nanoparticles, you are able to combine a treatment by the encapsulation of anti-cancer or antiviral compounds and the follow of the treatment by using imaging. So as a conclusion, I hope I have convinced you that the field of the nanomedicine is growing now very fast and very importantly. And what are the future challenges? The future challenges are certainly to develop multifunctional nanodevice. You understood that with high drug loading to avoid toxicity and without burst release to be more efficient. Also, you know that actually in the pipe of the pharmaceutical industry, there is some dry. There is a diminution of the number of chemical entities which are discovered. But the new drugs which are discovered are generally very specific of one signaling pathway, which is good from the point of view to avoid toxicity. But most of those drugs will induce very rapidly some resistance mechanism. In the field of the cancer, I have shown you one example, but also, and I had not the time to do that, in the field of the infectious disease. And I am convinced, ladies and gentlemen, that the use of the galenic approach, of the nanotechnology approach, will allow to fight, to overcome those resistance mechanisms. And finally, because the huge progresses made in the field of the material science, chemistry, and physicochemistry of colloids, you are now able to design, to use nanotechnologies and to design nanodiagnostics. This means nanoparticles with imaging and therapeutic possibilities. And I would say as a conclusion that the way to give is probably as important as what is given. And I would like to acknowledge all the people of my lab who collaborated more or less to one or another illustration I have given to you, and also the ERC, which has paid a lot to develop this type of research. Thank you very much for your attention.
Thank you, Professor Patrick. Now, last but not least, Professor Mustafa Qasim, who is going to deliver a speech about stem cells, basic biology, and clinical application. Please join me in welcoming Professor Mustafa Qasim.
Yeah, thank you very much. I'm really delighted to be here, and I would like to start by thanking Professor Ismail Sirajuddin and his wonderful team, really, of organizing such a wonderful event, and also of giving the opportunity and the empowerment of young people to be exposed to such wonderful science. So actually, I would like to dedicate my presentation to the courageous young people of Egypt who have been a great source of inspiration, not only for myself, but also for all the people in my lab. So I would like really to take you through 20 minutes of quick journey through the field of stem cells. And I would like to start by showing this slide, showing the background of my work. As a clinician, I and my colleagues are faced with this tremendous clinical problem of having increasing number of patients suffering from chronic degenerative diseases. And this is actually represent a major part of our clinical practice today. And this includes heart failure, liver failure, diabetes, and so forth. And unfortunately, we don't have treatment for these conditions. We have symptomatic therapy that can keep these diseases under control for a number of years, and we can't avoid the ultimate demise of the patients because of these therapies. But we have also a proof of concept that it is possible to treat these diseases, for example, by organ transplantation. But we are facing the challenge that there are no few organs. For organ transplantation, you need to use immunosuppressive therapy with its severe side effects of infection and probably development of neoplasia. And also there is an alternative where we have also a proof of concept that it is possible to generate tissues and functional tissues through transplantation of cells. And this is in particular for transplantation of hematopoietic stem cells and re-establishment of a functioning bone marrow organ in the context of chemotherapy of hematological malignancies. But as I mentioned in the beginning, what I'm going to concentrate upon today is the use of stem cells for treatment of non-cancer diseases, chronic degenerative diseases. So just a step backwards, what are these stem cells? You've heard them a lot this morning. So these stem cells, they are cells that are having this wonderful ability to differentiate to whatever cell type we have in the body. We have around 120 different cell types, and these stem cells, they have the capacity to do this differentiation. But at the same time, when they differentiate or when they embark on this differentiation, they have the ability also to keep one copy of themselves inside the niche or the microenvironment inside the tissue, and this is called self-renewal. And this is also behind the concept that these stem cells, they are responsible for tissue regeneration. And there is tissue regeneration in our body throughout life, but as we age, this regenerative capacity decreases. So where can we get stem cells right now? There are principally two sources of obtaining stem cells. One is called embryonic stem cells. These are derived from early post-fertilization organ. It's called the blastocyst, as you can see here. And this is, we get these blastocysts from supernumerary fertilized eggs that are generated in IVF protocols. And since 1999, there has been developing methods to isolate, to isolate a multi-talented group of cells inside these blastocysts. They are called inner cell mass. And these inner cell mass, they have the potential to differentiate to whatever cell types in the body, at least theoretically. So now we can isolate them and we can establish them in culture and study their phenotype and try to direct them to a particular tissue that we are interested in. The other stem cell types that we can work with and we can have is called adult stem cells. It's a misnomer, as you can see here. It's better to call them tissue-specific stem cells because irrespective of the age of the donor, we can obtain these cells from tissues. And also during the last 10, 15 years, there have been tremendous progress in development of methodologies to obtain these cells from tissues and establish them in culture. So what is the conceptual framework of the current situation in research of a stem cell? So the idea is that we can establish ex vivo cultures of stem cells in vitro, and then a lot of laboratories around the world are trying to say, how can we direct the differentiation of these stem cells into a particular tissue of interest with the aim of using them for clinical application. But actually, under the way, we can get also enormous insight, in particularly regarding the basic questions of biology, which is how this fertilized egg, which is the ultimate example of a stem cell, differentiate to all this multitude of different cells and tissues we have in the body. And this is actually a corner, the main question in developmental biology. Now we have a way to address developmental biological questions in vitro using human cells. This is tremendous importance. And at the same time, it also, stem cells provide tools for pharmaceutical industries to identify novel targets for therapy. I can't really tell you a lot of things about stem cells, so I decided to choose an example, a case study, to give you a flavor of how we deal with the stem cells in the lab and how we are trying to take them from the lab to the patients. I will give you an example of a stem cell type that I have been working with for many years. It's called the skeletal or mesenchymal stem cells, and this is one system of stem cells that is present in the bone marrow, and actually it was discovered as giving support to the principal system of stem cells in the bone marrow, which is a hematopoietic stem cell system. But also we realized that these cells, they are not only supporting hematopoiesis, but they are having a capacity to differentiate to other mesoderm-like lineage. So what are the characteristics of mesenchymal stem cells? They are present in the stroma of the bone marrow, but actually also in the stroma of other organs, including adipose tissue, joints, liver, pancreas, and even in the red bulb of milk teeth. Actually there, they participate in, or they can be used for generation of biodegradable teeth. But this is another story. So we know this, they are present at low concentration, 1 stem cells per 500,000 mononuclear cells in the bone marrow. There are some markers that can help us in identifying them, but these markers, they are sensitive. We can use them to identify these cells, but unfortunately they are not specific because they are expressed by other cell types. So usually we know that we have the right cells by showing that they can do what they are supposed to do. And what are these cells supposed to do? They are supposed to differentiate into cells of the mesodermal lineages, osteoblasts, adipocytes, and the chondrocytes. And in the literature, you will find some also controversial studies showing that these cells, they can cross the germline boundaries and differentiate either to ectodermal neuronal cells or endodermal, for example, hepatocytes. But these are controversial areas. This is actually to list the kind of research program. I have it in my lab, and it's dealing actually of taking a clear clinical question and going back in the lab, trying to find solution and hopefully go back to the patients again. From identification of homogeneous populations of cells, and we are using a lot of different technologies, including modern proteomic technologies. Been working a lot of how to improve the growth of these cells in vitro, in ex vivo culture conditions, and we are using genetic approaches, worked a lot with what's called telomerase to enhance the proliferation of these cells. I will concentrate on also in a particular studies that we are studying we are carrying out to control the differentiation of these cells into bone-forming osteoblastic cells. And also we have recent studies also related how to control the homing of these cells to injured tissues. We imagine that the best treatment modality for tissue regeneration would be actually to give stem cells like a blood transfusion. But this is also another very exciting area of research. I will concentrate on this idea. Here we have the stem cell. And now we would like to understand the grammar, taking these stem cells into bone-forming osteoblastic cells and not adipocytes or chondrocytes. How can we study that? The usual way of studying this, that we are in vitro, we are exposing these cells to a microenvironment conducive to differentiation to a particular lineage. And the main component of the microenvironment is based on growth factors. And actually we were in these studies examining the effect of a number of growth factors on the differentiation capacity of stem cells into osteoblastic cells. And we came into this situation where we found that one particular growth factor, which is the EGF, can induce osteoblast differentiation, as demonstrated here by the ability of the cell to form mineralized matrix in vitro. And this is a closely related growth factor with the BDGF. It did not do that. And actually, this was a good situation for us to try to understand what is the mechanism, what is the molecular mechanism leading to that the EGF can induce osteoblast differentiation and not BDGF. And as you can see here, these two ligands, they are used receptor tyrosine kinase kind of signaling, because when you do Western blotting and you immunoprecipitate all the tyrosine kinases, you can find that there are a big similarity. So actually we collaborated with a proteomic group in our university led by Matthias Mann, and we used a wonderful global quantitative proteomic method. It's called the Silac method, and it's a fascinating method. I will just mention it briefly. This method allows the labeling of control cells or EGF-treated cells or BDGF-treated cells with different isotopes of a particular amino acid. So when you run a mass spec, you can distinguish the proteins, whether they are derived from the control cell lines or the EGF or BDGF-treated cell lines. And then you can mix the samples, and this is decrease the sample variation between sample to sample. And then you can isolate, in this case, the receptor tyrosine kinase signaling proteins, and you run the mass spec. And as you can see here, we were able to identify over 90 tyrosine kinase signaling molecules, and as expected, most of them were similar in the EGF and BGF treated cells, but there were some differences. And we used a bioinformatic approach to find why were these differences, and we find that both ligands induce similar signaling pathway here, but with the BDGF there was another signaling pathway championed by BI3 kinase, and this was responsible for the differences in the biological outcome between these two treatments. But actually, these studies led us to the idea that maybe we should do a global knockdown of all the kinases that are present in the mesenchymal stem cells and looking at the outcome, because then we can get a dynamic readout. And we did it actually in two ways. I don't have time to present one of the ways, which is the siRNA knockdown of all kinases. This was one method. The other method that I am showing to you here, that we screened a chemical library of small molecule kinase inhibitors. We screened 80 different small molecule kinase inhibitors, and the readout was the differentiation capacity of the cells as determined by the expression of an osteoblast-specific enzyme. It's called alkaline phosphatase. And as you can see, this screen led the identification of one particular kinase, which call it KIX. And this is when we studied it more details, and it shows it increased the differentiation of a number of different cell strains of mesenchymal stem cells in vitro. And as you can see here, this is in the down panel. This is in vitro mineralization of the cells, which is also enhanced. And we combine these in vitro studies with in vivo studies by treating the cells with the kinase inhibitor and then implanting them in immune deficient mice and determining the amount of bone forming, which was much more in the case of the treatment of the cells with the KIX inhibitor compared to the control. And then also in a bone regenerative model, when we implanted these cells in a calvarial defect model of mice, the KIX-treated cells, they formed more bone than the vehicle-treated cells. So now we have a molecule where hopefully can we use to target the mesenchymal stem cells in vivo to enhance bone formation. So this is just a flavor of the kind of research we are doing. You can read much more about the original data in our publications. Now the last part of my talk will to tell you actually where do we stand regarding the use of stem cells in therapy. I would like to start by saying that currently there is no standard therapy using stem cells for these chronic degenerative diseases. So all the therapies that are being carried out are kind of clinical trials, most of them phase 1 or phase 2 clinical trials. And I know that there are some people say, okay, we can use the stem cells for treatment right now, but this is completely wrong. And usually there are a lot of commercial organizations trying to tell the patient that they can treat them using stem cells, and they usually advise the patient to avoid them, to avoid this organization. So stem cell therapy should should be carried out within academic institutions interested in clinical trials. But here also there are two concepts right now we are using for using stem cell in therapy. One of them, which I started by saying that we can replace tissues, and this is I will call cell replacement. So the idea here, for example, you take stem cells, you differentiate them to insulin-producing cells, and to inject them into diabetic patients to treat diabetes. So this is one concept. The other concept, I will call it stem cell-based humoral therapy. One of the greatest discoveries about the biology of stem cells is that these cells can produce a lot of biologically active molecules that can modulate immune system, they can modulate inflammatory process and promote tissue regeneration. And actually a lot of good results that we are getting from the use of stem cells, in particular the mesenchymal stem cells in therapy, are based on using of, for example, in treatment of ischemic heart disease, is based on the ability of these cells to produce a biologically active humoral substances, enhancing tissue regeneration. I will give you some examples related to cell replacement. Here, one example, the cells that I'm working with, they are able to form bone, so we can use them coupled to a scaffold, as you heard, in treatment of bone defects. And here is, as you can see, this is complicated fractures, and you can get healing after combining stem cells with a scaffold. And you've heard this very wonderful example about the use of stem cell, corneal stem cells for the treatment of corneal obesities. And actually this is an example where stem cells can be used as a medicine for the poor and not only for the rich part of the world, because as you know, this corneal obesity is very common in Egypt and in India. And here is just wonderful results of re-establishing transparent cornea after corneal stem cell replacement. We are working with because this is a nano session, so we are working with the iNano Center in Denmark to try to couple stem cells with nanoparticles and the scaffold. And the idea, if the patient has a defect and would like to fill it with bone, we can get a kind of a scaffold made, but in particular shape suitable for this defect. And the idea then to have this functionalized the scaffold, we will culture stem cells on the scaffold, and this scaffold will have nanoparticles containing, in this particular study that we carried out, siRNA to direct the differentiation of mesenchymal stem cells. And actually we have just published a proof of concept of this possibility, that we have this scaffold, which half of it was functionalized with the use of an siRNA controlling the differentiation or enhancing the differentiation of mesenchymal stem cells into osteoblasts, and the other half with an siRNA, enhancing the differentiation of mesenchymal stem cells in the adipocytes. And we implanted this functionalized scaffold in immune-deficient mice, and we were able to show that, as expected, we can form a composite scaffold made of 1 part bone and 1 part adipose tissue, which is a proof of concept that you can determine the lineage and the fate of mesenchymal stems in vivo based on this functionalization of the scaffold. I was fortunate to be involved in the first clinical trial where human stem cells were implanted in the brain of patients with cerebral hemorrhage. And the idea here, as I mentioned, a humoral therapy. So we know that mesenchymal stem cells, they can produce a lot of substances that can enhance tissue regeneration. And we added a layer of sophistication upon that because we engineered mesenchymal stem cells to produce a particular substance called GLP-1, and GLP-1 is an anti-apoptotic in the neuronal cells. So the idea is really to implant mesenchymal stem cells. There is this genetic GLP overexpressing together with alginate beads so that the cells can be attached to them, and to implant them with what has been termed as a tea bag. So when the patients come to us with cerebral hemorrhage, we remove the bleed, and then we implanted our genetically modified cells for 4 weeks. And this is the idea that to enhance tissue regeneration. And as you can see here is actually, this was phase 1 clinical trial. So it was a safety trial. It was possible to do the procedure. There was no transformation events of mesenchymal stem cells implanted in the brain. And actually, we observed some improvement in some patients, but of course, now we are moving into phase 2 clinical trial to prove the efficacy. So I would like to say here, what is then my biovision for linking science into society? Actually, after spending 3 days in this conference, I would like the youth of Egypt to have a novel revolutionary manifesto that the people that are coming to power in Egypt, whether presidents, ministers, or high officials, should be engaged in the biovision. They should believe in the message, and it should be obligatory for them to attend this meeting. So this is my message to the young people of Egypt. I really believe that with such a courage and the self-confidence that I experienced from all the young people in Egypt, I'm sure that their creativity will prevail, and I am sure that science and rationality will prevail Egypt one day. So thank you so much. And from our website, you can get a lot of details about the research that I referred to. So thanks again.
Thank you, Professor Mustafa. We have time for 2 questions.
My name is Abdurrahman Mahmoud, a student in Alexandria Faculty of Medicine, and I want to ask Dr. Hassan Azazi about the diagnosis of hepatitis C virus by using nanoparticles that it changes its color from red to blue when it is mixed with the RNA of hepatitis C virus. So how can we make sure that this change is due to the RNA of hepatitis C virus, not any RNA of any other virus? So is this mechanism can make interpretation of results between viruses or not? And thank you.
Thank you for your questions. As I mentioned, I was a little bit hurrying to go through the presentation. We use oligotargeter, which is a short DNA sequence. It is not a primer, it is not a probe. That is, we have done a great deal of work of bioinformatics to come up with this sequence that is highly specific for the conserved region within the 5' UTR region of the HCV genome. We have done, of course, a comparison study and clinical analyses in comparison with many other viruses and other RNAs. So we verified that this is really very specific to HCV, and we have done this in comparison with existing commercial HCV real-time PCR assays. Thank you.
Yes, I have 2 questions. The first for Dr. Patrick. And it's about, does using this biodegradable nanoparticles affect the recurrence of the cancer like hepatocellular carcinoma or cancer in pancreas or something other like that? And my second
Excuse me, I didn't hear your question. Could you repeat?
Yeah, okay. My question about using the biodegradable nanoparticles in treating cancer like hepatocellular carcinoma. There is a fiction, how does it reduce the rate of recurrence of the cancer, or there is no relation between it and the...
You mean just nanoparticles without... Yes, of course. When we are doing preclinical experiments, you know, on animals, of course, I didn't present it because we had no time, but we always are testing the activity of drug-unloaded nanoparticles and also a physical mixture of drug-unloaded nanoparticles and drug as a free molecule. And we have no improvement. Of course, normally the drug-unloaded nanoparticles have no activity, and we have confirmed that in the experimental model preclinical. But of course, clinically, you cannot test in the cancer field a placebo nanoparticles without drug for ethical reasons that you can well understand.
Yes, my second question is for Dr. Qasim. And
we said one question only.
Yeah, one question from Dr. Patrick and the other.
We said one question only, and we are right now at
about using mouse embryonic fibroblasts instead of there is a publication of the newest publication for you and your group, and about the best type of bone marrow cells that can be used, yellow or other type. Thanks.
I am sorry.
Yeah, I think to move into therapy using stem cells, we should avoid the contact between stem cells and any animal product. So you are absolutely correct that there is a huge interest in replacing the feeder layers, where we use sometimes to grow human embryonic stem cells with what's called the serum-defined conditions to be able to generate clinical quality grade stem cells for therapy.
Thank you very much. I have just one announcement before closing the session, that two of the top 10 next presentations will be discussed in East Exhibition Hall now. Please join me in thanking our distinguished panelists here for their valuable information and presentations. And now you can go to the break. Thank you very much.